Welcome back. In this second part, we will learn about artificial hybridization and apomixis. Let me first specify the objectives. 1. State the purpose of artificial hybridization. 2. Describe the steps involved in artificial hybridization. 3. Learn the different techniques of emasculation. 4. Explain the distinctive features of apomixis. 5. Distinguish between the three types of apomixis. 6. Specify the formation of parthenocarpic fruits. And 7. List the advantages of apomixis. So, artificial hybridization had become the key method of crop improvement, and the vast majority of crop varieties have resulted from artificial hybridization. Now, this is a process of producing hybrids through controlled pollination and fertilization between two genetically different plants. And some of the major objectives of hybridization are to combine desired characters of different plants into a single plant. And as stated earlier, it is a popular technique in order to create new improved crop varieties. And it also is a means to enhance food production. So let's study the stepwise process of artificial hybridization. The first step is selection of superior parents with desirable characters. In fact, selection of parents is the basic step in a hybridization program, and often, more than anything, determines the success of the breeding program. In the second step, the selected parents are made to self-pollinate for several generations in order to obtain the desired characters in all purity and consistency. And the third step is the careful removal of anthers before its maturity without affecting the female reproductive organs. And this process of removal of anther is called as emasculation. The emasculated flowers are then enclosed in suitable bags in order to prevent contamination. And this process is hence referred to as bagging. In cross-pollinated crops, the male flowers can also be bagged in order to maintain the purity of pollen grains to be used for crossing. Now, there are various techniques to do emasculation, such as hand emasculation. In this technique, the anthers are removed by using forceps in relatively larger flowers. The second step is called as suction method. Now, this is used in cases of smaller flowers where the anthers are removed by a rubber or glass tube attached to a suction pump. And anthers may also be killed by giving hot water or cold treatments. Or dipping of anthers in alcohol is yet another technique to remove the anthers. And the most effective method is inducing male sterility in the plants. Well, let's move on to the next step in hybridization. So, in the next step, the desired pollens are dusted manually on the stigma of the female parent, which marks the actual hybridization event. And on subsequent modifications, the hybrid seeds are produced from the parent plant. And such seeds are collected, tested for their performances by growing in the field. And if these seeds pass the test, or if they perform better than the existing parental plants, such seeds are allowed to be released into the market for commercial use. So with these steps, the plants are improved for food, fiber, fuel, and other economic services. Now, we will go into a certain basic understanding of some special modes of reproduction in plants. The phenomenon is known as apomixis, where apo means away from and mixis means act of mixing or mingling. Apomixis is considered as a form of asexual reproduction in which embryos are formed without fertilization. Hence, the embryos or the seeds which are produced by apomixis are the exact duplicate copies of their mother plant. And this occurs in plant species where sexual reproduction is either suppressed or absent. Apomixis is said to be of the recurrent type when the embryo develops from a diploid egg without fertilization. A diploid MMC or the megaspore mother cell forms a diploid egg by mitosis or in the absence of meiosis. And the resultant plant obtained from this type of apomixis is a diploid plant. Now, such kind of apomixis is prominently seen in onion and grasses. Differently, in non-recurrent apomixis, the embryos develop from the haploid egg without fertilization. And the haploid egg are formed from the MMC through meiosis as usual. And by consequence, the embryos formed in this way are haploid embryos. And finally, in the adventitious embryony type, the embryos develop from one cell or a group of cells of either nucellus or the integuments. And the embryos formed in this way are all diploid embryos. Now, this kind of adventitious embryony is most prominently seen in citrus and mango plants. Such a process of the development of more than one embryo within a single seed is called as polyembryony. So, apomixis has certainly proved advantageous to the plants. This is a rapid method of reproducing genetically uniform plants, and it's also beneficial in areas with all female population. And most importantly, apomixis had been widely induced in hybrid seeds in order to overcome segregation and to save the expense of buying fresh hybrid seeds every year by the farmers. An apomictic hybrid seed can be used year after year. Finally, let's get a brief idea about the formation of fruit in banana. Now, in a banana, an ovary directly ripens to form the fruits without fertilization of the ovules. Hence, the fruit becomes seedless. And the formation of fruits without fertilization is called as parthenocarpy, and such fruits are called as parthenocarpic fruits. And this kind of the phenomenon occurs naturally in plants such as banana, in pineapple, and also in figs. However, parthenocarpy can also be induced by the applications of plant hormones such as auxin and gibberellins. The seedless fruits which are easily available in the markets these days are commercially of value, as the consumption of seedless fruits is easier or more convenient. Yet, I would like to conclude here with the emphasis that all scientific practices must be ecologically sustainable. So with this, I come to the end of the second video on artificial hybridization and apomixis.